all the living things are made of cells. We cannot see cells with our eyes. We need a microscope to see them because cells are very small. Cells are like building blocks of the living things. They do different kinds of activities that the cells need to live. Animal cells and plant cells have 9 structures in common. They have a nucleus that controls everything that happens inside the cell. It's like a brain, and it also contains genetic information. Both have a cell membrane that separates the inside of the cell from the outside of the cell. They have cytoplasm, that is a liquid substance inside the cell where the other organelles float. They have mitochondria that works as a digestive system. It takes in nutrients, breaks them down, and produces energy. They have ribosomes that produce things that the cell needs to live, like, for example, proteins. They also have an endoplasmatic reticulum that is like a highway inside the cell. It transports different kinds of substances through the cell. They also have the Golgi bodies that are like post offices. They pack up things and ship them to other places. They also have the ribosomes that recycle waste products that are inside the cell. And they both have back walls that store food. Main differences between animal cells and plant cells are, for example, the shape. Animal cell has an irregular shape and the plant cell has a regular shape. Animal cell has centrioles. Centrioles are involved in the division of the cell. And the plant cell has the chloroplast that produces the food that the cell needs to live. And the plant cell also have the cell wall that helps the cell to maintain its shape. And finally, they both have back walls, but they are different. Plant cell has a big back wall and animal cell has little back walls. And these are main organelles of the cell and main differences. In the first session, I have planned 3 different activities. The first one is a warming up activity. I want the students to remember and revise the content of the topic. The second activity is a label activity. The students should be able to label the different organelles of the cell. The third activity is a compare and contrast activity. The students should be able to see the differences between both cells. In the second session, I've planned 2 different activities. The first one is a fill in the gaps activity, and I want the students to connect the names of the structures with their functions. And the second activity is a matching activity, and I want the students to match the shapes of the different organelles with their names and their functions. And this is all I have planned for these 2 different sessions.